Hi everyone, thank you so much for coming to my talk. My name is Kristen Marhaver and I'm so sorry that I'm here just as my voice and not actually in person as a human. I would have really loved to be there, especially for all the conversations in the hallway, all the support that's happening, all the brainstorms. So if you are moved by this talk, if you want to get into these species, if you want advice on larval propagation, if you have advice for me, please reach out after the talk. I'd love to hear what you're up to and compare notes. So before we get going too far, I want to thank everyone that I've been able to collaborate with over the past few years. And the people listed here in the top right are especially thanked in the context of this talk for participating in the gonochoric propagation project, either with a spawning observation or funding or help diving. So welcome to Curacao. If we go diving right where that yellow arrow is pointed in the water, this is what the reef looks like. One of the big advantages we have in Curacao is that the reefs are near shore and they're still in very good shape. And that means that we can make some pretty effective progress in spawning observations and larval propagation by having such convenient access to these animals. In fact, in the 90s, there was already a tradition of really successful coral spawning research happening at CARMABI. Manfred van Weghel spent a bunch of time figuring out when the hermaphroditic corals in the Montastria annularis complex spawned. We now know those as Orbicella, Franksii, Faviolata, and annularis. And I just wanted to give some attention to Manfred's work here so that we could appreciate the amount of effort that he went to with his team in order to figure out spawning times of these hermaphroditic corals. So in 1991, Mumford set up a 260-meter long underwater trail, marked 284 different corals. Divers monitored from 6 p.m. to 1 a.m., and there were about 75 divers involved in this monitoring effort. I love that they know exactly how many corals there were, but not how many divers there were. So this is the tradition of coral spawning research at CARMABI. By the end of the 90s, it sounds like it was a little bit easier to predict coral spawning. So Mark just brought this t-shirt back from his parents' house a few weeks ago from Coral Spawning 1999. And he said that if you bought the shirt, then you got free dive tanks and there was a barbecue and free beers after the dives. And it was a pretty big island-wide party, not only to appreciate coral spawning, but to continue to collect spawning observations. So I was lucky to join the coral spawning operations of CARMABI in 2007, and that was particularly when we were working on figuring out how to keep larvae alive so that we could study their behavior and their needs. As we got better and better at raising each one, we had a little bit more time to go watch for the next species that we might see spawning. In 2011, CARMABI's coral spawning predictions were about 10 coral species from August, September, and October. Fast forward 11 years, we've got 20 coral species. They're spawning in 7 or 8 different months. I also want to acknowledge Val Chamberlain and her team based in Curacao because she's done a tremendous amount of work to expand our understanding of spawning times in hermaphrodites, especially a bunch of the brain corals, and spawning times in some of the gonochoric corals that we're going to talk about next. So without Val, this graph would only be like half as big. And what's cool is that about half of the species that we're watching spawn now are gonochoric corals. So let's spend the rest of the talk talking about them. When Munford and his colleagues were out looking for coral spawning, they were looking for this. The massive boulder corals releasing these egg sperm bundles in this big mass spawning event, 1 night a year, 2 nights a year. But the more that you watch underwater in the dark, the more you notice these other quirky forms of spawning. Here is a pillar coral male spraying sperm into the seawater in big wispy clouds. And here is a pillar coral female spraying a bunch of eggs into the water in a big cloud. So gonochorism just means that male and female individuals are separate. You don't have hermaphrodites releasing eggs and sperm. You have colonies releasing either sperm or eggs. This group of corals is very understudied. They can be fairly patchy, they can be rare, they can be highly disease prone. So all of that can make them more complicated to find and to breed. But there are also a lot of gonochoric corals that are quite stress tolerant. They can be weedy, they grow in marginal habitats, in disturbed habitats. So there is a real potential to use this group of corals in restoration if we put in the effort to decode when they spawn and then how to propagate them. I've also found that some of these species actually grow incredibly well, robust, fast, happy. So as we've been following this group of corals for years and years now, we've made some progress. You saw the Dendrogyra pillar coral spawning in that last slide. We've spent about 10 years watching Dendrogyra spawn. Here is a 1-month-old in the middle of the screen eating coral food. On the right is a bunch of 2-year-old Dendrogyra colonies celebrating their second birthday. And then on the left is a screenshot showing the motility of Dendrogyra sperm after it was thawed back out after cryopreservation. We still have a ton of work to do before we can reliably fertilize gonochoric eggs with cryopreserved material, but we made it past this first step, showing that they can survive freezing and thawing. There's a ton more work to do there, and I'm really excited to try to push forward on that in the next few years. So that's a tricky one. Dendrogyra is endangered, it's rare, it's disease prone. And so is Dickosenia stokesii, the pineapple coral or elliptical star coral. Mark and I were working on figuring out Dendrogyra spawning for a number of years, and we decided we wanted to do something even trickier. And this was sort of the holy grail because there was only one spawning observation recorded, and that we know of, from Lori Richardson in Florida. We spent hours and hours diving, trying to find this thing spawning. Finally, Mark saw one Dickosenia spawn, and then we knew when to dive and kind of what to look for. So in 2017, we set out on this quest. Why did it take so long to figure out when they spawn? Well, this was what we were looking for. That's it. That's what we were looking for. You could be looking straight at it and not see it. The females didn't make it a lot easier. Here's a female spitting out some eggs. Some of them are a lot more dramatic than that, for sure, but this is why we only had one recorded spawning observation in the Caribbean for so long. You have to really, really know what you're looking for and really concentrate to see it. Once we discovered what we were looking for, instead of taking Manfred's approach, diving 1 or 2 or 3 nights with 75 people, we took 2 people and dove about 70 times. And so over the past 5 years, we've accumulated this pile of spawning observations. Now, what's very cool about Dickosenia is while the hermaphrodites spawn in a very narrow window, say maybe nights 4 to 8 after the full moon, Dickosenia seems to spawn whenever it wants. These are observations from September, October, November over the past 5 years. If you line them up end to end, you actually go from the end of September to the end of November. And we honestly still don't know where the start or stop is. We just got a great 0 in August, but that still leaves us like 4 or 5 weeks to figure out when this actually starts. At first, we couldn't find when this happens, and now we seem to not be able to find when it doesn't happen. So in those many attempts at finding spawning pineapple corals, we managed to make some progress in propagating them as well. Here are embryos going through development, and then juveniles growing up to about 2 years. Just through the CARMABI tradition of spending a lot of time in the water, being curious about spawning times, hustling to try to discover another spawning time for another understudied species, we've made a bunch of progress in propagating a bunch of these different gonochoric corals. But we're not the only ones working on this. I also want to highlight some of the progress that's happening region-wide because this is becoming an increasingly collaborative effort. Carrie O'Neill at Florida Aquarium Center for Conservation is propagating Meandrina meandrites as well as a bunch of other coral species, and we were really excited to see her grow Meandrina juveniles, knowing that we were hot on the trail of that quest. It's an honor to get second place to Carrie, and I think she's going to be talking later in the session about all of her awesome progress. In the Dominican Republic, the crew from Fundamar has been growing Dendrogyra on underwater nurseries for the past few years, and there's also been some great progress made in propagating them by Carrie and her team at FLAC, as well as the folks in Florida at Keys Marine Lab and Fish and Wildlife and Mote Marine Lab, etc. Also in Florida, the Figueredo Lab has been working on Montastria cavernosa and made some awesome progress in figuring out the conditions that help their juveniles grow and survive. There's an awesome team of people that's now monitoring Dickosenia in Nicaragua, which I'm so excited about. Val Chamberlain and her team based in Curacao have made a bunch of progress studying Siderastria and Montastria cavernosa. And then finally, Stephanostenia has been monitored over the years by folks in the flower garden banks, and then more recently people have been doing some larval propagation experiments with them as well. So this is just to show you that this is really a region-wide effort, And these species aren't so incredibly intractable that you can't join in as well. It's becoming more and more popular of a group of corals to study, and the more people that get into this, the better. I just want to sort of transition from these individual species stories to sharing a little bit about the best practices that we have discovered in the process of trying to grow all these different species. The most important thing with these species is you have to really get to know your population, not only when they spawn, but who's a male and a female, what they look like right before they're going to spawn. You've got to do some trial and error to find the sites that have good fertilization and good genetic diversity. Then you just got to put in your time underwater, just dive over and over and over. And there is a role here for spawning in aquariums, in ex situ systems, but we like to take some inspiration from Manfred, who brought his 75 people underwater for 7 hours, and we like to usually make it inconvenient on us instead of inconvenient on the corals. Principle 2 is handle with care. These gametes are being released free, not in bundles, and so if you can get them mixed underwater, that often helps get higher fertilization. You want to minimize the amount of shear force and abrasion that they experience. You want non-latex syringes because latex is toxic to sperm. And then even if you're doing everything the same, you will get a lot of variability in fertilization. So you have to really make sure your methods are spot on so you know that you did everything you could and the crappy fertilization is not your fault. Then principle 3 is actually also handle with care. As you're settling and growing out these species, you've got to keep them really clean. We found using a variety of micrograzers makes a big difference. Also feeding your juveniles, but watching out that you're not feeding pests and causing a big pest outbreak at the same time. One of the really important things about working with this group of corals is that there's an opportunity to make a big difference in their genetic conservation, even if you're only growing dozens or hundreds of individuals. To give you an example, there are dozens of Dendrogyra genotypes left in Florida, but there are now hundreds, if not thousands, of Dendrogyra in cultivation between Curacao and the Dominican Republic and Florida. So just to zoom out a little bit, a bunch of the rules of hermaphrodites, especially when they spawn, don't apply to the gonochores, but you have many days or weeks to figure it out, and once you get them settled and starting to grow, they're often quite robust. So there's a real potential to use these species in restoration, and I would encourage anybody who's kind of curious to just pick one that you love the most and give it a shot next year and see what kind of progress you can make. It's a really fun group of species to work with because any progress feels really thrilling and exciting, but also it's a fun group of species to work with because they keep us very humble. To give you an example of that, Dendrogyra, we thought we kind of understood its spawning times, but a couple of years ago, Mark and a colleague were out collecting samples for genetic analysis in January and February, and when hit with a hammer, a bunch of Dendrogyra colonies spawned. So a few hours too early and a couple of months too late. What are they doing spawning in February? We don't know. Do the females spawn in February? We've never gone to look. So they certainly keep us very humble. I will say that I was skeptical that this was spawn until Carrie O'Neill confirmed that her Dendrogyra on the other end of the region also angry spawn in the winter months when they've been cut with a saw. So this is a real thing. They somehow are making sperm in this whole season that we've never studied for their reproduction. So there's a lot more work to do, and that gives us, both urgency and opportunity. There are still hundreds of coral species with absolutely no reproductive data at all and no spawning observations. And we also have a really limited amount of time to get the coral genetic diversity left on Earth into younger and fitter and happier colonies. We're missing basically 30 or 50 years of juveniles. And so all the old colonies that are getting hit by disease and senescing, we have to get their genes into younger corals as quickly as we can. We have a lot more natural history to do, we have a lot more progress in breeding that we need to make, and we need to fund all of those things. We really can't just fund 2 or 3 groups to work on 2 or 3 species with 2 or 3 methods. We've got to fund lots of different folks to work on lots of different species. In other words, every coral species needs a champion. Let's diversify, pick the weird one no one's figured out before, and let's go figure out the propagation of all of these other species as well. So to learn more, you can go to my website, marhaverlab.com. I have links to these 2 talks, which talk a lot more about the specific spawning times of Dendrogyra, Dichocenia, Siderastria, Meandrina, and I would love to hear your feedback if you have ideas or you want advice about any of this. So just a big thanks again to everyone who collaborated on this and all the other projects that we've been working on this past year. And I hope you have a wonderful week at Reef Futures, and thanks so much for being here.